Thanks, Charles. Charles, can you see my screen okay? So what we're going to do today is to take a look at a fairly broad landscape of genetic issues around hereditary cancers and how those interact with central nervous system tumors. So as most of you know, tumors can be grouped into various categories, and for the purposes of this talk, we would group them as sporadic, familial, and hereditary. Sporadic, of course, is what we deal with for the most of our work. Those are not due to a single gene defect that's inherited through families. Familial refers to a parent clustering of tumors, but again, not due to a single gene defect. We often see skipping of generations with familial tumors, and then we get to hereditary tumors where indeed there is a gene that is inherited down through the family. Typically, we see affected individuals, at least carriers, of course, at every generation. And basically, if you look at the profiles of these three buckets of types of tumors, about 75% of cancers are truly sporadic. We'll see a parent familial clustering in about 15% and 10% of cancers, which, if you stop to think about it, is a very large number of tumors across across the landscape of the incidence of cancer. 10% of cancers have a hereditary single gene basis. And you can see some of the features of the differences is here. We'll just focus on the right side. And basically, this is about 10% of cancers. They're called syndromic because there are clusters of various types of tumors that occur in these patients. The patients tend to be younger than sporadic cases. They tend to have bilateral or multiple tumors. And again, the risk is monogenic. Now, as you'll all recall, what happens is that there's a single gene of the two. One is mutant. It goes down through the generations. And of course, there's a 50-50 chance in each generation of a particular offspring having acquired that mutation from their parents. But as seen in the lower right-hand part of the screen, cancer only occurs when the second copy of the gene is lost, and then that's when the cancer occurs. So that's the difference between the carrier state, where every cell in your body has the mutation, versus developing a tumor where the second copy of the gene is inactivated. So basically, here's another way of looking at the same thing. In sporadic cancers, there's about an equal representation of driver mutations, meaning those mutations that actually lead to increased mitotic rate in the tumor, between tumor suppressor genes and oncogenes. Oncogenes, straightforward, everybody knows this. They're activated by mutations. Tumor suppressor genes stop a variety of tumor type phenotypes in cells, and they are inactivated by mutations or various genetic events, and therefore lose their ability to suppress cancer. In hereditary cancer, the picture is extremely different. Here we see that almost all hereditary cancers are due to variants in tumor suppressor genes. And that's a very interesting phenomenon. There are about 80 to 100 known genes with about 15 syndromes, and the vast majority of those are indeed from tumor suppressor genes that are inherited from family generation to generation. Haploinsufficiency is the phenomenon that a single copy, normal copy of the gene, that's in all cells in an individual's body. They've got one mutant inactivated gene, they've got one normal gene, but that normal gene can't keep up with with the normal cell function. So haploinsufficiency says that if you lose one copy, somehow that begins to drive the cell towards more cell divisions, which then increases the risk of developing the second mutation, which is also known as the second hit. This is a double negative phenomenon where you are inactivating a brake on cell division, and therefore cell division becomes more common. That's a couple of examples there are BRCA1 and 2. That's the hereditary breast and ovarian cancer genes. NF1 is another well-known example to this audience. The reason that oncogenes are so poorly represented in hereditary cancers is because we believe that what happens is that if you inherit a oncogene mutation, that is embryonic lethal. So that individual actually never shows up in the family because they can't develop to delivery. And there are 5 counterexamples to the fact that oncogenes are not seen in hereditary cancer syndromes. They are shown there in the bottom of the slide, and it turns out that all of those mutations are very specific mutations in those 5 genes that are called hypomorphic variants. And what that means is that they are fairly active as oncogenes. They're weak oncogenes. Those particular mutations are not the genes, but the particular mutation in those genes. And so the reason those aren't embryonic lethal and occasionally come through, ALK, for example, is the cause of hereditary neuroblastoma. It's a point mutation in the ALK gene, which is a very weak mutation and therefore is not embryonic lethal. These are really fascinating concepts. So we're going to take that a little bit deeper of a step here. We're going to talk a little bit about what this looks like as you go across clinically. So basically, this is what hereditary cancers look like. We're going to start over on the left-hand side where a mutation is inherited from family member from generation to generation. This is what we say it's germline. The mutation is present in every cell in the patient's body. They get it from the parents. The gametes, 50% of the sperm and 50% of the eggs carry the mutation because the other copy, of course, is the wild type copy. And this is constitutional, meaning that it's in every cell in the body. And sure enough, if you look for that variant, the variant allele fraction is 50%. The number of copies that come off the sequencer, 50% of those copies that come off have the mutation or the variant preferred term, 50% of them don't. And so that's what that looks like. And basically, if you have a tumor, the tumor would also have a variant allele fraction of 50%. And so that's what that would look like. Now, when you get to the next stage, we call these de novo tumors, which mean that they arise for the first time in the individual that's identified as having the cancer. So your dad and mom, one of them didn't have cancer that then got passed on. This is like, you're the first lucky person. That can happen at either the prezygotic or the zygotic stage. If it's prezygotic, that's a mutation in a sperm or an egg cell. That's more common than you would think. It's called a gonadal mosaicism. And if that happens, then it turns out that every cell in your body, it's going to look just the same as if you'd inherited that from a parent with the carrier status. And basically what happens in that situation is that the variant allele fraction in blood is 50%, just as if it was inherited from a germline, from your parents. If the first cell stage, the zygotic stage, gets the mutation, and that's very common, for example, we think in NF1, probably 50% of patients with NF1 are de novo, then you still have that 50%, that 50-50 variant allele fraction. When we get over to post-zygotic, now this is maybe at the 8-cell stage or even the trillion cell stage, you see that if early on this mutation occurs, then we only see it in a fraction of cells in blood. And if it's really late, which is the case with sporadic cancers, the blood fraction is 0, but the tumor contains lots of the mutations. Now, this concept of variant allele fraction is very important. I'm going to spend a couple of minutes with this because I don't think a lot of us get to sort of work through this. Imagine a situation where you have a gene, you've got 2 copies of it in peripheral blood lymphocytes. There is no variant. You're wild type for that. So you've got 1 wild type copy, 2 wild type copies, that's 100% of your genes, so your variant allele fraction is 0. If you have a BRCA1 mutation where every cell has 1 abnormal gene and one wild type gene, 1 mutant gene, the variant allele fraction is 50%. And so you've got one, you see the math. It gets a little trickier when you look at the tumor because tumors are composed of normal cells and tumor cells. So in the case where the tumor cell fraction, 80% of the tumor in the sample that you send to the genetics lab is composed of tumor cells, well, now we've got our wild type cell with at 20%. This should say wild type on the right there. You've got 20% of 2. Well, no, I'm sorry. You've got 20% of cells that have 1 wild type, 1 mutant copy, and 80% of the tumor cells still have that wild type and mutant. So again, the variant allele fraction is 50%. It gets a little trickier when you get to sporadic tumors because now what you can see is that you've got 20% of the cells in this case are wild type for both copies of the gene. So in essence, what you can see here, it's very straightforward, is that the wild type copies in the tumor and the non-tumor cells gradually get diluted by the increasing numbers of cells that in the tumor. And if you get up to the place where the tumor cells actually both have the same mutation in them, and there's some mechanisms where that occurs, you can actually get a variant allele fraction of 80% in the tumor cells. So let's do a case. Here is one of Charles Cobbs's cases. This is a lady that he and I saw together before I left. 42-year-old woman, she's got a little unsteady gait, gets a scan, has something that looks a whole lot like a cerebellar hemangioblastoma. Charles took that out. The pathology was hemangioblastoma. So the question now is does this woman have sporadic hemangioblastoma? It was the only lesion we saw in her cerebellum, or does she have von Hippel-Lindau syndrome? This is an example of how the variant allele fraction analysis can be so powerful. So this was her family tree. Didn't look like there was anybody that had anything that looked like VHL syndromic tumors. But the problem was that she had a cyst in her left kidney, and she had multiple lesions in her liver, and both of those can occur in von Hippel-Lindau. Spinal cord imaging showed no Peel hemangioblastoma. But this is tricky, right? So does this patient need to go through screening for the rest of her life because she's got von Hippel-Lindau, or does she not? Well, the way you handle that is you send off genetic testing. And so we sent the tumor, that's the somatic, it's so called in this report from Tempus, and we sent blood for germline testing. And the report on this woman is that they didn't find anything looking like VHL in either one. Now, the trick here is that when you turn the page, they report a variant of unknown significance in the VHL gene. Variant of unknown significance means, in the opinion of Tempus, this particular variant at the 313th nucleotide changing an A to a C, that did not meet criteria for a pathogenic change. And this was seen in tumor somatic. And the variant allele fraction was a meager 14%. So what do we make of this? Well, what we did was we did an in silico analysis of that particular variant. And it turns out that it's located in a mutational hotspot. And so if you actually look at the computer evidence looking at protein function and structure, it's actually very suspicious, but not strong enough evidence perhaps to call a straight out pathogenic variant. Well, now can we bring in that 13.8% variant allele fraction to help us? So here's the math on this. Notice over on the left-hand side that the tumor cell fraction in this particular patient is 70%. Was 30%. 70% of the cells in the tumor in the hemangioblastoma were deemed to be normal. And so if you do that same analysis that we just did a moment ago and you look at this, it turns out that the predicted variant allele fraction is 14%, and there it is. So that's very, very strong evidence that what this woman actually has is a sporadic a sporadic mutation in that tumor of the VHL gene, but she does not have von Hippel-Lindau syndrome, and therefore we don't have to worry about her family, her children. We don't have to worry about screening for the rest of her life. So what we did was we took that case and we reported it to the National Library of Medicine database of all mutations, and they have a big panel that gets together and look at this, and they deemed that to be a pathogenic mutation. And so now that particular base change is in fact classified as a mutation. So if you look across all hereditary cancer syndromes, this is what things look like. If you look at the known hereditary syndromes, the red ones have a central nervous system component to them. And basically what I've done here is to classify them by the type of molecular alteration that we see underlying the syndrome. I'm going to skip over this slide, but basically we think of hereditary cancer in a brain tumor patient when we see young age at diagnosis, certainly if there were to be multiple tumors, if there is a tumor that is highly suggestive like choroid plexus carcinoma of belonging to a known syndrome, those are red flags. I'll finish with three very quick examples. Cowden syndrome, this is classic imaging for dysplastic gangliocytoma of the cerebellum. You can see those so-called tigroid streaks in the cerebellar lesion. It's non-enhancing. This is, in an adult, is pathognomonic of Cowden syndrome. In children, not so. Cowden syndrome has a number of tumors, but the thing that we get from a CNS standpoint is Lhermitte de Clos and a big head. In Lynch syndrome, which is colon cancer without polyps, we see glioblastoma, but there are a number of other tumors that we can see in the syndrome as well. This is a very interesting area that I'm doing research in because it turns out that the CNS tumors often don't show some of the molecular features of mismatch repair genes, and that's an extremely interesting area of research. Here's familial adenomatous polyposis, in which we see medulloblastomas, or very rarely some other CNS tumors. And it's interesting, that's wingless signaling, and that's known to be the first class of medulloblastoma subtype. Lee-Fraumeni syndrome, TP53, they get tumors all over the nervous system, but the classic tumor, choroid plexus carcinoma. If you see choroid plexus carcinoma, it's almost certain that that patient has a congenital TP53 mutation. And then my last slide is a picture of our book cover, which will be out at the beginning of April, co-written with a very famous genetic counselor, Robert Resta. And that brings my presentation to a close. Thank you.